Hi everyone. In this video, we're going to start talking about lipids and lipid synthesis. So we saw how fats are broken down, fatty acids. Now we're going to see the whole repertoire of lipids. It's actually a fascinating class of molecules that all cells depend on. It's extremely varied. I mean, the lipids come in the most flavors than any other molecule, so it's quite something. Where does that fit into our whole anabolism or our whole organizing scheme of metabolism? It's clearly an anabolic process. So we're going to see how we start with small oxidized precursors and put to use energy in the form of ATP and reducing power in the form of NDPH. This is where all our NDPH is going to go, or I mean a significant fraction of NDPH goes to actually make lipids. It's a major sink of NADPH, lipid synthesis, that is. So today, in this video, we're going to cover just the overview of the various lipid structures. Okay, so it's extremely, as I said, it's a very diverse class of molecules. So we're just going to try to put it together and put them in different boxes so at least we know what we're talking about. What do they do? Of course, they provide energy, as we saw with fatty acids. When you break them down, you get a ton of ATP. It's the energy stores. But the message we're going to try the point we're going to try to make with this video is that actually they have many other purposes, functions in the cell. So lipids, of course, they provide structure. This is it's the fundamental building block of all membranes. Membranes are diverse. In the sense that they contain different kinds of lipids, which endows them with different kinds of properties. Lipids, something we don't think a lot about, but it's extremely important, also have a signaling role, so specific lipids, as we will see. So they come, as I said, in many different forms. So the general lipid categorization is shown here, where we have two major giant groups, ketoacyl lipids, and isoprene lipids. We're going to go through them one by one. We're used to, so far we have seen really one, fatty acids, which is a part of ketoacyl lipids. So fatty acids, we've seen them before. That's how they look like, right? I mean, there are variations in the side chain, length, and you can have double bonds. You can also have branches, but that's pretty much it. Now, we've also seen those. So what happens to fatty acids when they are esterified with glycerol? They can make glycerol lipids. So the backbone here is glycerol, and the side chains are fatty acids. And you can have one or up to three on any given glycerol backbone, as we have seen. Now we start seeing new things that we have not seen before. So we have glycerol phospholipids. So, or more general phospholipids. Very important. These are actually the building blocks that we are talking about in biological membranes. So you have the same glycerol backbone, and you have two fatty acids esterified as before with a typical triglycerides and glycerol lipids. But and one of them, a third one, is actually there is a phosphate. And then there is some hydroxylated compound of some sort. So it could be ethanolamine, choline, serine, or carbohydrate even. Phosphatidylcholine is probably the most common, probably by far the most common glycerol phospholipid in biological membranes, in both, let's say, yeast and animal and human membranes, on the plasma membrane especially. So here we start seeing some variation again. We have a phosphate here, and then we have some other small compound usually. We're going to see in more detail how these compounds look like. They're pretty small, but we're going to discuss the synthesis of these phospholipids when we talk about how they're made. Now we branch further out, and we don't have glycerol anymore as the backbone, and now we're talking about sphingolipids. And sphingolipids look like this. We have sphingosine, they're built around sphingosine. And sphingosine, you can see now the acyl side chain. It's not actually acyl at this point, but you see this hydrophobic side chain. It looks very similar to the side chains of the fatty acids we have seen. And that's the end part of sphingosine, however, is different. So now we have the hydroxyl groups, and we have an amino group. And now, depending on what gets attached to those groups, you have a whole variety of sphingolipids. Okay, so this is a pure sphingosine, and then if you stick onto the sphingosine backbone, so sphingosine now would be the backbone, and if you put a fatty acid, you have a ceramide. If you have a phosphocholine and a fatty acid, you have a sphingomyelin and so on and so forth. And they also, you can have phosphoethanolamine and the fatty acid. And you keep building. Okay, you can have some. If you just have one sugar, then you have a cerebroside. If you have a variety of sugars plus sialic acid, let's say, then you have a ganglioside. So this, the last two, cerebrosides and gangliosides, are very common in brain cells, as the name implies. For example, in ganglia. So what I want you to appreciate from this, and there's going to be more variation as we go on, is again, this enormous variation. You start building different chemical groups. So you have an extremely variable and broad range of chemical species. You can sample a lot of chemistry with lipids and a lot of different conformations. And that's really what and why cells love them so much. Imagine if you stick those things on your membrane. I mean, the membrane all of a sudden is decorated, and those will face on the outside. It's decorated with a whole different complexity. It allows you to see and be seen in many different ways. Then, now the backbone gets something really very different from glycerol or sphingosine. So now we have the sugars. You have here a disaccharide, and of the various hydroxyl groups, let's say, of the sugar, you can just stick various things on, right? All kinds of stuff. So those are the sacharolipids, because again, the backbone is a sugar. Lastly, these are the most bizarre of all, polyketides. They're really natural products that are synthesized in bacteria for the most part, sometimes in fungi, but mostly in bacteria. And their synthesis is very similar to the way fatty acids are synthesized, which we will see shortly in another video. But they adopt so they are built, all these enzymes, it's a multi-enzyme complex. They are built almost like a factory, an assembly line, and you make this diverse, very diverse species. And here is one drug that we're going to see later in another video, later in the course, rapamycin, very famous drug. It's used in cancer as also an immune suppressant, and more recently in aging studies. So this is a polyketide. It is a lipid. It's made by it's made in streptomyces by this assembly line sort of synthesis. So these are all ketoacyl lipids. A complete different class, which also starts, however, from acetyl-CoA, as you will see. Is the isoprene class. So that's the fundamental building block of isoprenes, of isoprene, and that gives you, why do we care about this? It gives you sterol and cholesterol, for example, and other hormones. It's of obvious health significance. Prenols, so there is a sterol ring, okay, so with this building block, the isoprene unit, you can make the sterol ring, and then you can decorate in many different ways, and you can get all kinds of stuff. We're going to see some of the structures. So this is very prominent in fungi and animals. Plants do a lot of prenols instead, and they kind of look like this, and you can put them together in more complex structures too. So we'll conclude this video and at this point.